Now that you've watched an overview, let's test the information you gained from this video. The final product of an expressed gene can be which of the following? Find all the correct choices. Once you're ready, hit play on the recording to check your answer. Good job. Every single one of those items can be just a product of transcription. Let's break down these steps of protein synthesis. The first is transcription.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
Now let's look at translation.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anticodon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated. Three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
How does this process differentiate between prokaryotes and eukaryotes? In prokaryotes, as you can see here, everything happens in the cytoplasm of the cells. If you note, RNA processing does not occur in prokaryotes, while in eukaryotes, Transcription and processing occurs in the nucleus, and translation occurs in the cytoplasm. Let's look at an overview of transcription. It is fairly similar to replication. An RNA polymerase that moves in the same direction from DNA polymerase 3 in the 5' to 3' direction binds to the sequence in DNA known as a promoter. The RNA polymerase will then move down the sequence of DNA to be transcribed and creates an mRNA product. Now let's look at how transcription begins. The promoter region is known as the Tata box. It is a specific sequence of DNA that precedes the gene. These promoters can be found in a variety of places throughout the genome. Transcription factors will bind the promoter once the RNA polymerase II binds to the factors. The initiation complex is complete and the transcription will proceed. As transcription proceeds, this process is called elongation. It is very similar to how DNA is replicated. There are 3 primary differences. RNA is a single-stranded molecule, the sugar in the backbone is different, and thymine is not an RNA base. Instead, uracil is paired with adenine. This process proceeds until a termination sequence is reached. Once mRNA is made, it is modified before translation can occur. The first modification is an addition of a 5' cap and a poly A tail. These modifications help guide the ribosome to the correct coding region. Each rough mRNA has introns and exons. These introns were originally thought of as junk RNA, but researchers have found these introns can drive different aspects of translation, such as quantity of product of protein. These introns are removed from the complex using spliceosomes. Once these enzymes remove the introns, exons are sent to the cytoplasm for translation. Another role for introns is to separate the different domains of proteins. These domains are simply different regions of the same protein. The introns can help ensure that each region is folded appropriately. The big question for translation is how is each amino acid assigned? Sets of 3 bases on the mRNA sequence are called codons. Each of these codons are specific for an individual amino acid. The first 2 bases are the most important bases in the sequence. The last base is known as a wobble base and can be different without impacting the amino acid that will be added to the sequence. Let's practice a question with regards to this. Write down the sequence above and work through the questions. Bring your answers to class for discussion. When you are ready, hit play on the recording to continue on. The mRNA is translated into a protein using a ribosome. This used to be called the rRNA. Can you name each of the parts shown here? Pause the recording and try to name them. When you're ready, let's check your answer. Good job. How close were you to getting all the parts named? The E site stands for the exit site, and this is where the tRNA will leave after it has added its amino acid to the sequence. The P site is the peptidyl tRNA binding site. This is where the amino acid is added to the sequence. The exit tunnel is for the amino acid strand that's being formed, and the A site is where the tRNA will first enter the ribosome. There's an mRNA binding site that helps feed the mRNA sequence between the large ribosome and the small ribosome. Now that we are familiar with the ribosome, let's look at the tRNA. These are named for the shape of their sequence. This picture shows the different levels of RNA folding. The anticodon at the bottom of the tRNA is what pairs with the codon on the mRNA. This picture shows how the amino acids are attached to the tRNA. These amino acids will separate from the tRNA when they are in the ribosome to join the forming amino acid chain. Now let's look at one cycle during translation. Remember, this can occur many times until a full protein has been coded. This process continues until a stop codon is read. The ribosome, amino acid sequence, and the mRNA will all separate from each other. Just like in DNA, it is possible for mistakes to be made. These mistakes can have varying levels of consequences. On the right-hand side of the graphic are what is known as nucleotide pair substitution. This is where one base is altered in a sequence. This can have 3 types of downstream effects. The first is a silent mutation, where the change can't be seen. A missense mutation will change the amino acid within the sequence, like we see in sickle cell, and a nonsense mutation can prevent the protein from being folded at all. The right-hand side of the graphic shows us what happens if a nucleotide is inserted or deleted. This causes what is known as a frameshift mutation and can create dramatic effects. As you can see in the picture, several amino acids can be altered when a frameshift occurs. As I just mentioned, this is our timeless example of sickle cell. As you can see, sickle cell is a result of a nucleotide substitution. As you remember from Chapter 5, this small changes causes a dramatic change in the shape of red blood cells. Let's tie this back to our laboratory exercise. These examples below of the pig and the tobacco plant demonstrate the ability of genes from one species to be expressed in a different species. This is possible because of which property of the genetic code? Let's wrap up this lecture with a few questions. Our first question is, which of the following is the best example of gene expression? Pause the recording, and when you are ready, hit play to check your answer. Good job. B is the correct answer because fur color is a result of gene expression. The rest of these are changes into the genes themselves. Now I want you to take this list below and decide which of these items pertain to replication, transcription, modification, or translation. And each one can be used more than once, and bring it to class for points of discussion. Now that we have finished Chapter 17, you should be able to do the following objectives. If you can't, please see me so we can work through some of these items. As always, we'll be doing some exercises in class to help cement these objectives. And here's some supplemental information to help you understand the material. If you have any questions, please see me in my office hours or in class, or email